Treatments for deafness focus on replacing missing stimulation. Generations ago, people used ear horns to capture sounds and direct them into the ear more loudly than usual. Nowadays, we use hearing aids, which are basically mini audio amplifiers, as a microphone picks up environmental sounds, amplifies them, and places the sound directly into the ear canal. By providing louder than usual sounds, the hearing aid effectively overcomes the mechanical problems inherent in conduction deafness. However, it cannot help as effectively in sensorineural hearing loss, which is when hair cells are damaged or missing. While the hair cells may be lost in sensorineural hearing, the electrical excitability of the auditory nerve remains intact. This is when we use cochlear implants to directly stimulate the auditory nerve with electrical currents. While cochlear implants do not restore normal hearing, they provide stimulation the brain can interpret as sound. Brain imaging indicates that the stimulation activates the auditory cortex in a tonotopic manner, which means they allow good but not perfect perception of tonal aspects of speech. Studies confirm an increase in speech perception with continued use of cochlear implants. The best linguistic outcomes occur when implants are received early in life, and the success of these implants seems to be mainly due to the cleverness of the brain, not so much the implant itself. Mammals traditionally have been viewed as incapable of regenerating hair cells, yet we now know that, similar to fish and birds, the supporting cells packs around the hair cells of the mammalian organ of Corti do remain capable of dividing and differentiating into hair cells in adulthood. Human clinical trials of ATOH1 and other drugs are currently underway, hopefully leading to an effective treatment for sensorineural hearing loss. So vestibular perception relies on hair cells to track position and motion of the head. When you turn your head, take a turn in your car, or bounce on a boat, your continued awareness of motion allows you to plan future movements and anticipate changes in perception due to the movement of your head. It is the vestibular system that informs the brain about gravity, movement, and the position of the body in space. Too much of the stimulation can make you sick, though. The inner ear system senses gravity and acceleration. So the vestibular system is an inner ear system that encodes the orientation and acceleration of the head in 3 axes, which are crucial for the sense of balance. The axes are Y, which stands for yaw, the x-axis, which is pitch, and then the z-axis, which is roll, which will be further explained when we get into the semicircular canals, which are the most obvious parts of the vestibular system. They are fluid-filled tubes oriented at right angles to each other. They are oriented in 3 different planes in which the head can rotate: nodding up down, or pitch, which is the x-axis, shaking your head side to side, yaw, or the y-axis, and tilting your head left or right, or roll, which is the z-axis. The 3 canals are connected at their ends to the utrosil. Lying below is another fluid-filled sac called the saccle. The canals work together to provide accurate tracking of the head's rotation. The ambula is an enlarged region of each semicircular canal that contains the receptor cells, or hair cells, of the vestibular system. 3 of the hair cells are embedded in the copula, which is a gelatinous structure. Orientation of the steroclia determines the direction of the mechanical force to which it is particularly sensitive. Otoliths, which increase the sensitivity of the receptors in the utrocyte and sulcus in movement. The vestibular system evolved from the lateral line system, which is a sensory system commonly found in fishes and amphibians. It consists of an array of receptors alongside the body that informs the animal of water motion in relation to body surface. Nerve fibers from the vestibular portion of the vestibular nerve synapse in the brain stem are important in motor control and posture. Nerve fibers from the vestibular receptors enter lower levels of the brainstem and synapse in the vestibular nuclei. Some fibers pass this structure and go right to the cerebellum, however. The vestibular system is involved in the precise control of eye movements, as evident in the vestibulo-ocular reflex, or the VOR. This maintains gaze on a visual object despite movements of the head. Functions accurately even when the eyes are closed, showing that vestibular inputs, not visual ones, control the accuracy of gaze. Some forms of vestibular excitation produce motion sickness, however. Motion sickness is the experience of nausea brought on by unnatural passive movement. It is caused by too much strong vestibular stimulation, and it's caused especially by movements of the body we cannot control. Sensory conflict theory is a theory of motion sickness suggesting that discrepancies between vestibular information and visual information stimulate food poisoning and therefore trigger nausea. This means that an example would be that your vestibular system signals the bouncing of an airplane, yet your visual system sees the plane's interior as constant. So, chemicals in food are perceived as 5 basic tastes. Taste and smell, it's important to note, are chemical senses. So humans can distinguish 5 basic tastes: salty, sour, sweet, bitter, and umami, which is a savory, meaty taste. All 5 basic tastes can be perceived anywhere in the tongue where there are taste receptors. The sensations uniquely aroused by an apple or a steak are flavors rather than taste because they involve smell as we taste. This means if you blocked your nose, a raw potato would taste the same as a raw apple. Taste excites specialized receptor cells on the tongue. Taste buds are not the myrid little bumps on the tongue. These are papillae, which contain most of the taste receptor cells, and there are 3 types. The circumvallate papillae, which is located at the back of the tongue, and there are relatively few of these. The foliate papillae, which is located along the sides of the tongue. Again, there are relatively few. And then the fungiform papillae, which is located in the front of the tongue. There are hundreds of them, yet the numbers vary among individuals. They contain at least 6 taste buds each. Taste buds now are a cluster of 50 to 150 cells that detect tastes that are found in the papillae. At the surface end of the taste bud, you'll find the taste pore, which is the small aperture through which tasted molecules are able to access the sensory receptors of the taste bud. A tastin is just a substance that can be tasted. Different cellular processes transduce the basic tastes. So, salty and sour are evoked when taste cells are stimulated by simple ions acting on ion channels in the membrane of the taste cells. Sweet and bitter are perceived by specialized receptor molecules and communicated by second messengers. And at least 2 receptor proteins may be involved in perceiving umami. The taste of salty is due to the transduction of the taste of salt, or NaCl, mostly relies on ion channels. While other tastes are either appetizing, such as sweet or umami, or unpalatable, such as sour and bitter, salt could be both. When you have low concentration, salt is appetizing. However, when you have high concentration, it's unpalatable. The taste of sour just means the more acidic the food, the more sour it tastes. No one quite knows how sour tastings are detected. However, it seems hydrogen ions may play a role. The taste of sweets. Receptors are more complex than those involved with salty and sour tastes. It basically involves G protein receptors going to secondary messengers. These secondary messengers are then broken down into T1R and TR2. T1R taste receptors are proteins that, when particular membranes heterodimerize, form taste receptors for sweet and umami flavors. TR2 is just a family of bitter taste receptors. And then that sums everything up. Thank you.